Bismillahirrahmanirrahim. Today we are going to discuss the thermoregulation in living organisms. You know, in your syllabus you have to study the thermoregulation in plants and animals. First of all, we are going to start with the plants. In plants, you know, there are two problems which they face, either in low temperature or high temperature. When there is low temperature, the cyclosis, the movement of cytoplasm, that is disrupted, okay? And I mean the streaming does not occur. And if the streaming of the cytoplasm does not occur, it means that the molecule wouldn't be able to move from one place to the other. It would also mean that the enzymes wouldn't be able to move from one place to the other, and the cell would die consequently. But the plants are intelligent enough to produce the ice crystals around their cytoplasm in the plasma membrane. For this, they undergo an arrangement, that is what they increase the amount of the unsaturated fatty acids in their plasma membrane. You know, plasma membrane is formed of two major constituents, what the lipids and the proteins. Okay, if you have studied the fluid mosaic model, you might remember that. So in this way, when the ice crystals are formed around the cytoplasm in the plasma membrane, the cytoplasm enjoys the streaming movement or the cyclosis. Taken. So this is how the cells, rather the plants, they arrange to thermoregulate during low temperature conditions. The second problem the plants do face is what? The high temperature condition. You know that there is an optimum temperature at which the enzymes of the cells or the plants they perform their functions of catalysis. Suppose this is an enzyme. Okay, you know, enzymes are formed of amino acids. These are the globular proteins, and in their globular structure there is a catalytic site and binding site. Okay, and catalytic site and binding site, they form what? The active site. So active site is a particular part of a globular structure of protein. Suppose this is maltose. Okay, the plant cell requires glucose. Now, what is the solution? The catalytic site must break this maltose. Okay, and it wouldn't be able to do that if there is a high temperature. During a high temperature, what will happen? There would be the disruption of one part of the catalytic site is here, the other part of the catalytic site is here. There wouldn't be no more catalysis. Okay, if the plant has to carry out certain other biochemical reaction, its globular structure should be maintained. Its conformation shouldn't be disturbed. The enzyme should have its own conformity, so that there should be healthy catalytic site and binding site. So if the condition is this, what will happen? No enzymatic activity would occur normally, and the plant would die. Now, what is the solution? The solution to this problem is, as I always say, to every problem there is a solution. Okay, to every problem there is a solution. So when there is a high temperature, the plants, they produce certain proteins, and those proteins are what? The heat shock proteins. These heat shock proteins, they protect the enzyme and if the enzyme is protected, its globular structure would be maintained. And if the globular structure is maintained, what will happen? There would be optimum rate of metabolic process. Okay, and this means if there is no disorder, if there is no problem in the metabolism, what is that? That is healthy plant. So high temperature, but the heat shock proteins, they maintain the normal health of the cell and subsequently the plant. So this is how the plants, they maintain their thermoregulation in low temperature and high temperature. Thank you very much.